Okay, so now we have ATP, or we're moving on to metabolism. So we're going to talk about ATP, which is adenosine triphosphate. It is a primary source of free energy in living cells. The structure is a nitrogenous base, adenine, attached to the 5-carbon sugar ribose, which is attached to a chain of 3 phosphate groups. So it's O negative P dash O dash P dash O dash P dash O. On top of the P's is a double bond to O. Below the P's is a double bond to negative O, connected to a little pentagon with 2 hydroxyls at the bottom, connected to O at the tip, and then there's 2 rings with N's around it, connected to an H2N. Now we're going to talk about electron carriers, which are redox reactions, or coupled up reactions, compounds that pick up electrons from energy-rich compounds and then donate them to low-energy compounds. The electron carrier is then recycled. How they work: the compound accepts the electrons, so it gets reduced. The compound loses an electron, means it's oxidized. Electrons that pass from one atom to another carry energy with them. A reduced form of a molecule has higher energy. Electrons are said to carry reducing power. Then we have 2 types of reactions, so endergonic and exergonic. An endergonic reaction is any reaction that requires an input of energy. The change in free energy is positive. The products of the reaction contain more free energy than reactants, and the bond energy is higher. Disorder in the system is lower. So example is photosynthesis and the input of light energy. Then we have exergonic, which is reactions that release excess free energy as heat. Products contain less free energy than reactants. Bond energy is lower, or the disorder is higher, or both. An example is cellular respiration with the breakdown of glucose, which produces ATP and releases energy. Then there's 4 stages of cellular respiration, which is glycolysis, pyruvate oxidation, Krebs cycle, and electron transport chain. First, we are going to talk about substrate level phosphorylation. When ATP is formed through the direct transfer of PI, which is an inorganic phosphate, to ADP using an enzyme. This results in the generation of less energy than alternative. Oxidative phosphorylation is the alternative, which takes place on the inner mitochondrial membrane, and we will discuss that when we learn the ETC. Okay, now we have glycolysis. The products of it are 2 pyruvates, 2 ATPs, 2 NADH, and 2 H pluses. What happens is we start with a 6-ring glucose with a little one at the top. ATP turns to ADP, and then we have the same glucose molecule with a P attached at the top. Then we have ATP turning to ADP, and then we have the same glucose but split in a pentagon shape with 2 P's on each side with also a dot, which is called a diphosphate. Then it splits into G3P, so 3 dots, 1 P. What comes out of this is NAD plus turns to NADH, and inorganic phosphate enters, PI enters. Then we have 3 and P on each side. ADP turns to ATP, then we have 3 and 1 P on one side. ADP turns to ATP, and then we just have 3 dots, which is called a pyruvate. The input is 1 glucose, 2 ATPs, and the output is 2 NADH, 4 ATPs, and 2 pyruvates. The net is 2 ATPs. Then we have pyruvate oxidation. So first, the 2 pyruvate molecules formed by glycolysis in the cytoplasm are transported through the mitochondrial membranes to the matrix. So pyruvate oxidation happens in the matrix. So what it starts with is a pyruvate, CO2, which is the waste that you breathe out, exits, and then it becomes 2 dots. Then NAD plus turns into NADH, which is to be used later in the ETC, and it still becomes 2 dots, but now it's acetic acid. Then the enzyme coenzyme A enters, and it becomes 2 dots, and then CoA, which is coenzyme A. This is to be used in the Krebs cycle. Then we have the Krebs cycle, which occurs in the matrix of the mitochondria, and it is aerobic. So what happens is we start at an oxaloacetate, which is 4 dots. Then the acetyl coenzyme A enters, and the 2 dots enter, but the coenzyme A, it gets removed. Then it becomes a citrate, which is composed of 6 dots. Then CO2 exits, NAD plus turns into NADH, and it becomes 5 dots. Then CO2 leaves, NAD plus turns into NADH, and we have 4 dots. Then GDP and an inorganic phosphate turn into GTP, just like ADP, and also ADP and ATP are having something going on, and substrate level phosphorylation at the bottom. Then we still have 4 dots. We have FAD turning to FADH2. Then we still have 4 dots. We have H2O entering. We have NAD plus turning to NADH, which takes us back to an oxaloacetate with 4 dots. Then we have the ETC, which has lots of things. So what happens is that NADH will transfer electrons to the first protein complex, so NADH dehydrogenase, in the ETC, and so pumps 3 hydrogens per molecule. Then we have FADH2, which transfers electrons to the Q, which is the second protein in the ETC, and pumps 2 hydrogen per molecules. The inner mitochondrial membrane is impermeable to NADH, so NADH is created in glycolysis must be shuttled via a transfer protein to the matrix. In doing so, it becomes FADH2. Now we're going to talk about chemiosmosis. So it is the electrochemical gradient generated during electron transport that stores free energy. It's called The inner mitochondrial membrane is impermeable to H+ ions. PMF forces proteins through ATP synthase, and the energy from this drives the synthesis of ATP from ADP and inorganic phosphate in the matrix. 1 ATP is generated per protein pumped into the intermembrane space. After ATP is made, it is transferred through the mitochondrial membrane by facilitated diffusion into the cytoplasm, so it can do things like active transport. Then we have ethanol versus lactate fermentation. So the diagram is a pyruvate, CO2 exiting, which means takes it to 2 dots, which is an acetaldehyde. Then we have NADH from glycolysis turning into NAD+, which results in 2 dots again, and is ethanol. Because of the lack of oxygen and no ETC in bacteria, NADH gives electrons to aldehyde to keep glycolysis going with NAD+. Then we have lactate fermentation. So the diagram is a 3-dot pyruvate, NADH from glycolysis turning into NAD+, which forms a 3-dot lactate. Because of the lack of oxygen, the ETC is shut down. NADH from glycolysis gives electrons to the pyruvate instead of NADH dehydrogenase. So pyruvate oxidation and Krebs cycle won't happen because there's no pyruvate. The NAD+ goes back to the glycolysis to keep it going. Then we have the basal metabolic rate, which is the amount of energy needed to keep an organism alive. It accounts for 60 to 70% of energy used in one day. It is measured in kilojoules per square meter surface of body surface per hour. It increases from birth to the end of the first year, then gradually decreases. It is also affected by health and fitness. Then we have the photostem. So the photostem is complex of chlorophyll molecules, accessory pigments, and proteins embedded in the thylakoid membrane. There are 2 parts: the antenna complex, which is a number of chlorophyll molecules and accessory pigments, and then the reaction center. The antenna complex absorbs a protein and transfers the energy from pigment to pigment until it reaches a chlorophyll molecule in an area known as the reaction center. A transmembrane protein complex containing chlorophyll A, whose electrons absorb light energy and begin the process of photosynthesis. An electron is raised to a higher energy level. Electron is transferred to the primary electron acceptor in the reaction center. In photostem 1, a chlorophyll A is called P700. Absorption spectrum peaks at wavelength of 700 nm, or red light. Then we have photostem 2. A chlorophyll A is called P680. Absorption spectrum peaks at a wavelength of 680 nm, red light. Then we have the Calvin cycle. So there are 3 steps. 1 is carbon fixation, 2 is reduction reactions, and 3 is Ruby P regeneration. So we start with ribulose biphosphate, or Ruby P, which has 5 dots and a P on each end. Then we have, and beside that is a ×3. Then we have 1 dot CO2, which is ×3, but not connected in the line. Then 3 H2Os are added. And then we have 3 dots and a P on one end, which is PGA, or 3-phosphoglycerate, and this is ×6. And then 6 ATP turn into 6 ADP, and then we're left with 3 dots and 2 Ps on each end, ×6. Then 6 NADPH turn into 6 NAD+, and 6 inorganic phosphates, or PI, are exited. Then we are left with 3 dots and a P on one side, which is G3P, or glyceraldehyde 3-phosphate, ×6. And with another round, and then underneath that, it is left with 1 G3P. So with another round of the Calvin cycle, 2 G3Ps become 1 glucose. Then we have another G3P, which is 3 dots and then a P, ×5. H2O is entering, 2 inorganic phosphates are leaving, 3 ATP are turning into 3 ADP, and then we're back to the ribulose biphosphate Ruby P, which is 5 dots and 2 Ps on each side. So, yeah.